Welcome to Vision to Venture, the podcast where we talk to leaders in the tech world. In each episode, we chat with entrepreneurs, tech experts, and leaders within the tech space to hear their stories. So, if you're curious about tech, starting a business, or just want to hear inspiring stories, this podcast is for you. Make sure to follow and subscribe to never miss an episode. Let's get started. Welcome to Vision to Venture. Today, we are joined by Arvind, who is the co-founder and CTO of Tessel Bio. With a rich background in data science and AI from companies like Verily Life Sciences, DE Shaw Research, and Benevolent AI, Arvind has brought his expertise to the forefront of drug discovery. So we're really happy to have you on the podcast today, Arvind. How are you?
I'm doing great. Thank you for having me, Emily. It's great to be on the show.
Brilliant. Absolutely great. We're really excited to see sort of what we can get into today. So I think first things first, obviously in the position you're in, it would be really interesting to find out what initially drew you to sort of the technology and the life sciences sector.
Absolutely. So I guess like a lot of people, I had a lot of interest in math and that kind of technical stuff growing up. And the kind of natural extension from that was to physics and computer science. Certainly that's where a lot of my friends were going. And that's where the kind of peer pressure interest was pushing me towards. For better or for worse, I had a fantastic biology teacher in ninth grade who really showed me that biology was kind of beautiful and how complex it was, but also kind of importantly made me believe that biology could be engineered in the same way that physics could be used to, make chips, make circuits, make code. Biology could be built up in the same way. I have to say that at the time, I think that really was something I took on faith. She did a very good job of instilling that faith, but I don't think I had enough evidence to make that call. But it paid off. I got to college, and after years of telling my math and physics friends, hey, biology is just as cool, just wait and see, I actually got to see. I started working on something called synthetic biology, which is at core, how do you get cells to do the things you want them to do? And as I studied that and a few other things, I came to understand the kind of boundaries between different subjects in a very clear way. I'm sure you've seen before one of those explanatory videos where you start at the level of a human body and it starts zooming in. You get to the level of an organ, then you get to the level of the tissue underneath, and then to the cell, and then to things happening inside the cell. What I was learning at the time was kind of like that, but in reverse, from the most fundamental level up to the level of cells. I got to see how quantum chemistry fit together with the physics of molecules interacting, fit together with biochemistry, and finally fit together with what was going on within cells. And one of the nice things was I could kind of jump back and forth between those levels as I learned more and more. You're understanding how the different parts of a cell are interacting together. Maybe you want to make a change there. And you understand exactly what that change needs to look like. So you're able to dive down deep into the physics and the chemistry to see what do you need to do to make that happen. Once I felt that level of understanding and control, it was almost a foregone conclusion. This was what I was going to work on. There was no way I would be able to understand anything else as well as I understood this. And from that point on, I've been hooked. There's definitely more I can say, but at the end of the day, human biology is an incredibly complex system. And if you can actually break it down and build it back up in this way, there's nothing that's more satisfying. The impact on humans, frankly, is just the cherry on top of the pie. With all of that, there's nothing else I'd rather be doing.
Yeah, nice. I mean, going all the way back to school and even at university, I touched on a little bit of the biology side of things. It's always been the science that I've been kind of drawn to. I've never understood physics or chemistry or that side of things. It's always been biology. So I think it's absolutely fascinating that you've obviously taken, I guess what you could say is a big passion you've had ever since you were sort of very starting to learn that area of things, and kind of inspired you to actually start what you've done now. So in terms of what Tessel Bio are doing at the moment, what sort of is the company's mission in the drug discovery space?
Absolutely. We have a kind of specific right now mission and a bigger mission. I'll talk about both. The specific mission right now is to focus on making drugs for chronic diseases, particularly those in epithelial tissues. It's worth breaking both of those down a little bit because when people hear epithelial tissues, they think skin, and we're more than that. And chronic diseases may be a little obscure of a term. It's not clear exactly what you mean. So let me start with chronic diseases since that's the easier one to explain. Cancer is big on everyone's mind. Everyone, if they live long enough, get cancer. Cancer really sucks, and we'd like to find better ways to treat it. But it usually doesn't come out of the blue. Sometimes it does, but a lot of the time, there are things happening to you as you get older, ways in which your body's failing, not fatally, but definitely inconveniently and often pretty badly. And the poorer our treatments for those things, the more quickly and the more likely it is that we actually reach a cancer. Those are chronic diseases writ large. Ways in which your body fails, partially genetically motivated, partially environmentally motivated, that lead your body to deteriorate more and more until inevitably it ends up with cancer or another fatal disease. Now, as far as epithelial tissues, really your epithelium is any part of your body that directly interfaces with the outside world. Skin is a big part of that for sure, but you also have things like the lung. Anything you breathe has to get into your bloodstream somehow. The lung mediates that. You have your gut. Anything you eat has to get into your bloodstream somehow. The gut mediates that. You have your kidney, and you have a host of other organs and tissues in your bodies that serve as epithelial tissues. Putting these two together, chronic diseases on epithelial tissues, we think that we have an approach that works particularly well there. That all speaks to our particular mission, but we have a kind of larger mission, which is to make drug discovery more predictable. What I mean by that is, today, when a new disease pops up, when patients start having a new disease, work starts on the disease very quickly, very rapidly. There's a huge push, a huge effort to treat it, to cure it. But if you ask any scientist, any doctor, when will there be a drug for this disease? They have to say, I have no idea. That's how the process works today. You try a lot of things, and eventually, through a combination of hard work and luck, something is found, and a drug is built on top of that. We want that process to be a lot less open-ended and a lot more predictable. When a new disease pops up, we should be able to say, here's the approach we will take. It works eventually for all diseases, and more, we can actually tell you how long we think it will take for it to work. In the process, we would also like to bring down costs and the actual time it takes to get to a drug for a disease, and hopefully also make the drugs more effective. So obviously that's a big set of goals to have. We're early, we don't know if we'll succeed, but it's the vision.
Yeah, I think it's good to have lots of goals, and also very very interesting goals in terms of actually doing some real good as well. And obviously from I know a bit about your background. You've worked, like I said at the beginning, kind of at Verily Life Sciences, DE Shaw Research, and also Benevolent AI. So how has that experience, sort of working across these various roles in data science and also AI, actually shaped your approach to drug discovery and what you're doing at the moment at Tessel Bio?
Absolutely. So I think every company that I've worked at has had a lot of strong people and a lot of very well articulated and very well run processes. There's been a lot to learn from, for sure. There's also been a lot to learn from the blind spots that each of these companies has had. That's going to be true of any company you work at. I can only hope that in a few years someone will be able to say that about us. But I think I've taken just as much from the blind spots as from the strengths, and I've been able to put them together in a nice way. I will say there's a lot of organizational things to learn in terms of how these companies actually structure projects, structure their employees to work on those projects, how they manage the work that gets done. That probably applies kind of across not just machine learning, but any kind of software science kind of task. There's also a lot of lessons that we have learned about doing drug discovery in particular, and how machine learning in this space is kind of different from machine learning writ large. Happy to talk about that more if you're interested.
Yeah, definitely. This episode is sponsored by Enigma. Enigma help connect machine learning and software engineering talent with groundbreaking opportunities in the field of AI. So, whether you're a company searching for exceptional tech talent or an individual seeking that perfect role, Enigma is your go-to solution. Thank you to Enigma for sponsoring Visions of Venture. I mean, obviously your background is fascinating, and you've obviously got some, advanced skills coming from all these previous roles that you've had. And like you just mentioned, machine learning actually plays a really big role in the work you guys are doing. So yeah, it would be great if you could go into a little bit more detail about sort of applying that machine learning to drug discovery and what you're sort of doing in that area.
Absolutely. So I guess there's a few different ways to frame this. But at core, what we're trying to do at Tessel is to first pick measurements that you can take in a dish, that you can take in the lab, that are more predictive of what happens at clinical trial. This is important because, and this actually speaks to a little bit of how I got into this field in the first place. When I was in college, I heard a talk from a representative from Ernst & Young. And at the time, he put forth a figure. For every 5,000 drugs that starts off somewhere in the drug discovery pipeline, only one actually makes it all the way through, gets approved, and reaches patients. Now, I should say, even at the time, this was 2009, there were plenty of caveats around that number, whether it was accurate, whether it even mattered. Um, but that idea that you could have such a big discrepancy in how many things are tried and how many things actually reach patients really really stuck with me. I thought if you could make that ratio even a little bit better, then the drugs that actually did succeed wouldn't have to pay for quite so many failures. So already you can lower the cost of drugs, but also you could focus on trying things that are more likely to work and hopefully actually get to more effective drugs at the end of the day for patients. This was kind of what was motivating me. But as you can see from there, the big win is making sure that fewer things fail in clinical trials, because you put together all of the costs of everything tried before clinical trials and all of the costs of everything tried in clinical trials. It doesn't even compare. You could save tons and tons of money in the preclinical trial portion, and it would be a drop in the bucket. You really need to make sure that fewer things fail in clinical trials, and we wanted to push strong on that front. So again, one of our big efforts at Tessel is to try and pick measurements, ways to assess drugs early before you reach the clinic that are much more predictive of what will actually happen in the clinic. The second big push at Tessel is to find ways to computationally model those dish endpoints, those dish measurements, so that rather than going and doing 1,000, 10,000, 100,000 experiments in the lab to try and find the best drug, you can actually move that into the computer. You can say, what if I tried this drug? What if I tried this other drug? You can test all of those in the computer before you actually move to doing things first in the dish, then in a mouse, then in the clinic. And so it's all about first picking those measurements and then building machine learning models of those measurements so you can do what people call virtual screening, screen these drugs at scale. That is what Tessel is pushing to do. And we've come up with a number of ways to kind of make that practical, ways that we think the industry is at the moment undervaluing or not focused on.
No, that makes sense. And I mean, you've just gone into it a little bit there, but I think it would be good to go into it in a little bit more detail. Obviously, with what you're doing, you're combining sort of computer-based methods along with biological data to actually try and produce these kind of valuable results. So is there anything else that sort of goes into that and that makes it particularly difficult sort of in the drug discovery area?
Yeah, definitely. Maybe before I answer that specifically, it's worth kind of mentioning how we unite these two things in our mind. Many people might hear kind of the word model and assume you're talking about machine learning model. But if you talk to a biologist, that's actually not what they think. When they hear the word model, they think some kind of measurement that you can take, which is informative of something else that they care about. So what I was just talking to you about, the idea of taking measurements in a dish in the lab and hoping that that tells you something about what's going to happen in a human later on, that is a biologist's model. And at core, those two are really the same thing. A model at its core is just an approximation that you can more easily use than the real thing that you care about. And so Tessel, our goal really is just to build better models, full stop. Whether that's biological, whether that's computational, we want to build better models that you can use more easily, more quickly, and at scale. Now, why that actually comes out and why that ends up being hard in biology all comes down to one thing at the end of the day, data. No matter how hard we try, and we are improving over time, collecting data in biology is very time consuming, very difficult, and very expensive. I feel relatively confident saying that definitely not in the next decade, but possibly never will the volume of data in biology reach the scale that we talk about when we talk about things like LLMs. That's just not in the cards because of how intricate biological data is. Again, there's some caveats around that claim. Happy to talk about them more if you'd like, but that informs all of our design, all of the way we do machine learning, because we can't assume that data will always be plentiful, that we can build massive models to take advantage of that. We have to work within the constraint that data will always be scarce, and every piece of data has to be treated as very valuable to that end. There's a few things that that I at least have learned from my previous experiences that feed into how we kind of build around that constraint. Happy to talk about that more as well if you like, but that is really the core constraint we're operating under.
Yeah, sure. That makes sense. And I think, everything you've discussed there, it's quite interesting that you can kind of kind of umbrella the word models to cover both of them, and that made complete sense with the way you explained it to me. So that's great. And I mean, as well, I think you just mentioned it as well a little bit. So naturally, there are some sort of strict rules and regulations when it actually comes to the drug discovery side of things. So how do these kind of regulatory kind of constraints actually affect how computation and machine learning can actually be applied during drug discovery?
Absolutely. So I will say that our innovations, even when we're trying to make clinical success more likely, our innovations are all preclinical. They're focused on what you do in the lab before you get to even mouse trials, let alone human trials. What that means is that we are not trying to do anything to change clinical trials or to speed them up. We're just trying to make sure that we are less likely to fail them. Now, there are many, many ways drugs can go wrong. And for all the malignment that the FDA, that the regulatory scheme gets around the world, the regulatory structure we have today goes a long way towards preventing many of those mistakes. So credit where credit is due. They're obviously not perfect. There are times when they are too stringent. There are also times when they're not stringent enough. And there's definitely room to improve them. And maybe AI will play a role. We actually have some ideas around that. But that is not our primary mission, to improve clinical trials or to improve the structure of clinical trials. All of that said, I actually think that the regulatory structure makes the machine learning problem a lot easier for us. When you're a company making models that will ultimately serve some kind of consumer, there's a lot of customer discovery involved. What does the customer actually want? How will they use these models? What's the best way I can make these models for them? In our case, it's pretty clear what our goal is at the end of the day. We can haggle with the FDA to try and change the clinical endpoint they ultimately measure. But really, they are telling us this is how we will assess whether a drug works or not. Do better at that. That's such a clear goal, such a clear metric. And the clearer the metrics that you use, the easier machine learning will end up being. And so from our end, we actually really appreciate how much guidance, how much direction we get from this kind of regulatory scheme in drug discovery. And we think that's something that benefits us in a surprising way, in a way that other industries don't have.
Yeah, that makes sense. I think that's really interesting, actually. So thank you for going into a little bit more detail about that as well. And just onto something, I guess, a little bit similar is obviously you've got data collection side of things when it comes to drug discovery. And I think that's quite known to have its challenges. So when you're dealing with that, how do you sort of overcome challenges such as sort of the cost of things, the time that it takes to actually ensure that the process is efficient and, doing what you need it to be doing?
I'm really glad you asked that, actually, because I guess in line with what I was saying earlier about the data limitations being the clear thing that separates biology from other fields, this is our focus in a way that I think it isn't even for most of the other companies in our space. We want to build performant models, and we want to find effective drugs, yes, but we want to do it with as little training data as possible, just because there will not be that much training data. And that necessarily means we have to collect the most informative training data. We have two ways of thinking about that. One of them is what we call active learning, basically going step by step, not pulling in all of the data up front, but rather at every point collecting the data that we think will be the most informative, and actually letting our models tell us what data they will find most informative, what predictions they're the least certain about and would most benefit from actually seeing data for. Collecting that data, updating our models based on that, updating our understanding of the models based on that, and then repeating the process, asking them once again, now that you've seen that, what are you now most uncertain about? In this way, even though it will take more steps, right? You can't go out and commission lots of data at once, do training in one step and be done. You actually end up getting to a better endpoint with a more performant model at the end of the day, with much less data than you would have needed otherwise. So that's one of the big things that we push on. The other is actually focusing on what data we are collecting, not necessarily what kinds of inputs we put in, but what are we actually measuring? So I'll talk a little bit more about this. You could think that you have some set of cells in a dish, you're measuring something on them, and what you're really trying to do is find the best drug to treat those cells and ultimately to treat the human. So you could go through and you could try different drugs and see what each one does and train your model based on that and train it to predict what new drugs will do. That is the kind of active learning approach that I already talked about. But there's another step here too. What do you actually measure about the cells once you put the drug in? Not all data is created equal. You can measure what the cells do functionally. Do they form a tight barrier? Do they secrete anything? Do they change their shapes? All of these things. You could also measure what's happening inside the cells. What genes go up and down? How do the genes work together? Are there any chemicals that are produced that you wouldn't expect? Things like this. There's a million different things that you can measure about the biology once you do something to it. And you can't measure all of it. And most companies think that if you can't measure all of it, the best thing to do to achieve scale, to achieve real prediction, is to pick one thing about the cells that you can measure, and get really good at measuring that one thing at scale. We disagree. We think that the best piece of information that you can pull out at any given time isn't always going to be from the same modality. Sometimes you might want to know what two genes are doing together. Other times you might want to know which ones went up or down. Other times you might want to know something about the cells and how they work together. Your model needs to be able to tell you what modality is most informative at any given time, and you need to be able to go and pull out that information for your model to learn from. So while this does limit us in terms of the kind of scale we ultimately can achieve, or at least how quickly we can achieve it, we go after the information that's most informative, no matter the modality. And that's how we think about data collection.
Yeah, no, that makes sense. And obviously, you've got a process that kind of works for you now. I feel like from what you've said, you've made it quite effective for what you guys are doing. And obviously, you yourself have worked across sort of the entire stack of drug discovery. So has this actually helped you sort of to understand and be able to work across sort of the full spectrum of this?
Definitely.
Yeah.
So I think drug discovery is normally broken down into a number of subproblems. I guess anytime you have a field this big, you have to do that. There's no way you can kind of expect everyone in the company to look out at the whole thing. You need to be able to break it down so people can work in different parts. The problem arises when people pretend that these subproblems genuinely fit together no matter the circumstance. That assumption tends to lead to more difficulty than is needed, and it also leads to drugs that don't actually work as well as people want. I can give examples of what I mean by this, but one of the big benefits I've gotten from working across this whole stack, across the full spectrum, is I can see when the assumptions that are kind of baked into the field, baked into the field's DNA, if you will, actually apply and when they don't. And because of that, I can help us structure our own divisions according to the project we're working on. Maybe one division of work makes sense for one disease, but another division makes sense for another disease. I can talk about this a little bit more. I think it comes across a little abstract right now, but in drug discovery, there's usually 3 steps. The first is defining the disease. You might see a bunch of patients popping up with the same kinds of symptoms and say they all have the same disease. But really, that doesn't hold unless giving those patients the same drug leads to the same outcome. That's what we mean by a disease, that the same thing is happening to them and they can be treated in the same way. Otherwise, they have 2 different diseases that look very similar. From there, once you've actually defined which patients have the disease, you need to pick how you're actually going to reverse the disease. What biological knob do you want to turn so that their disease behavior will go back to looking like healthy behavior? Finally, you need to make a drug that will actually turn that knob for you. This is how people think about drug discovery today. But there's lots and lots of ways that breaks down. First, diseases don't have to separate into these clean little buckets. They can kind of meld along a fluid spectrum. You need to be able to say, this person has a disease that's relatively similar to this person, and we think that because they're more similar than 2 other patients, a drug that works for one is more likely to work for another. Or we might be able to get the same effect that we need with a combination of drugs, something like that. Then when you turn around to say, this is the one knob that we're going to turn, you have to understand that realistically, turning only one knob might not actually get you all of the effect that you need. It might also cause side effects that you don't like. You might need to turn more than one knob at once, even if that seems like a more complex thing to do. Finally, when you're working with drugs, you have to understand that one drug probably isn't only going to turn the knob that you want it to turn. It's going to turn other knobs too. And it's really, really hard to make sure that never happens. But maybe that doesn't matter that much. That doesn't matter all the time. It might be the case that for some diseases, it's really important for your drug to be very specific. It might be the case that in some diseases, one particular off target, right? One particular thing that your drug does wrong is very important, but a lot of them aren't. Understanding the details of these kinds of problems, when the drug specificity matters, when the drug strength matters, which knob actually does drive what you want, and which other knobs could be helpful versus harmful. Understanding all of these things holistically actually gives you a lot of power to do drug discovery more easily and more effectively. the final example that I'll give there is, imagine you're making a drug and you work really hard to make it specific. It turns the one knob it needs to turn 100 times stronger than any other knob. You might think, I've done as well as I can do here. I should move on. This is plenty good, right? But for some diseases, it might not be good enough. Maybe you needed 1,000 times stronger or 10,000 times stronger. But maybe you only needed that kind of specificity compared to one other knob that it accidentally was turning. If you could focus the drug on avoiding one of the off knobs, but let it be sloppy with all the other ones, then you could actually get the drug to behave exactly as you want without spending quite as much effort. This is a relatively technical point, but the idea is that you can see these ways to make your life easier and the patient's life easier as you do this work, if you have that whole vision.
Yeah, I think the technical side of things is actually really interesting. And I think a lot of people listening actually will be interested in that side of things. So thank you for going into a bit of detail about that. Yeah, it was fascinating. And just to kind of change it up a little bit, obviously, you're sort of the co-founder and CTO. And from that sort of perspective, I know that you guys are a startup at the moment. And can you sort of talk about sort of the difficulties of actually meshing the biochemists that you hire with the sort of computational engineers that you hire, and how you sort of manage that and how you work through that?
Absolutely. This is a point that's kind of actually very, very important to me. Maybe I'll step back for a second and give a little bit of context on how I think this fits into a bigger picture for us. So ultimately, I think when you're making models, when you're making approximations, right? There's a fun quote: "All models are wrong, but some are useful." I think the maybe more accurate version of that is all models are approximations. You can't help that. And it helps to know how your model is an approximation, where you can use it, when you can use it, and when you can't. And so when we think about kind of building models, there's three things that we need to worry about: making the models good, learning how to improve them, and knowing when we can and can't use them. These are kind of the three pieces that fit together. Hiring is actually one of the big ways we see to make the models good in the first place. When you hire groups of people who have a hard time communicating and thus collaborating because they speak different languages, they have different experiences, it becomes very hard for them to build models, because now you don't only have approximation errors in random places that you couldn't have predicted. Everywhere they work together, everywhere they kind of take their parts of the models and put them together, you're going to have gaps because the two groups didn't understand how to work together to close those gaps. And so in some sense, if you can't deal with meshing, as you said, biochemists and computational engineers effectively, your models are going to fail before they've even started. And they're going to fail for reasons that were completely avoidable, just because your two groups weren't able to talk to each other. This is something that I've seen happen at some of my previous employers, and it's had a really big effect on me. The solution that we have is essentially to guarantee that even when our hires don't have the exact same experiences, they at least have curiosity about the parts that they don't know. Ideally, you would have people who come in with experience across the stack: machine learning, biochemistry, molecular biology, all of these things. And for a company like ours, where we're relatively small, just 5 people right now, we can actually guarantee that all of our employees, in fact, do have experiences across the board. But that won't be possible to scale indefinitely. And we need to make sure that when we reach a point where that's not true, curiosity takes over. A genuine willingness to engage with people, to understand their viewpoint and how it's different from yours. I don't mean that in a kind of general way. It's easy to be open-minded and say, I will listen to you, I will hear what you have to say. But it's an entirely different thing to take yourself out of your comfort zone and understand that their workflow is so different from yours, that these biases, these good practices that you've built up over a career, no longer apply in their field. I'll give one particular example. The biggest difference between computer science and biology is the time scale. If you're an engineer, you write a piece of code, you maybe compile it, and then you run it. And in the old days, that whole process to run it and then see the result and then learn something from that would take maybe a few seconds, maybe a few minutes, definitely no more than a day. That's unheard of in biology. If you're running an experiment, it'll usually take a week, maybe a month, maybe a year or years. And when you're operating on that kind of a time scale, silly mistakes or lack of care will lead to much bigger and much more avoidable errors. And so biologists tend to be very, very careful about the kinds of work they do, the way they set up their experiments, in a way that computer scientists just don't intuitively understand because they haven't needed to. Now, machine learning has actually closed this gap in more ways than one. For one, machine learning models take a while to train. And so as a result, machine learning engineers have picked up some of that care in their own work. At the same time, machine learning has also made it possible to do some experiments virtually. And so biologists are able to get by with a little bit less caution now in their work. But there's still a pretty big gap here. And addressing that is honestly one of the biggest issues that we see. We don't have a great solution for it yet, but it's on our minds. It's something that we flag for all of our employees, and it's something that we're thinking about constantly. We are optimistic that we will be able to do better and better at this, but it's a work in progress.
Yeah, it makes sense. And yeah, you never really think about that in terms of how long sort of a biology side of thing can take. And you're right, versus a computer is completely different timescales, isn't it? So I can't even imagine what that's like, but it seems like you're doing a decent job at kind of trying to manage that. So I guess on to sort of a final question. We'll go into like go big or go home with this one. Just keen to know what your thoughts are. So how do you sort of envision sort of Tessel Bio actually influencing the broader field of drug discovery and sort of biotech in the future?
Absolutely. As I mentioned already, we want a world where drug discovery is more systematic, where the time and the money isn't really as limiting, isn't exorbitant in the way it is right now. That's also a world, ultimately with shorter time from disease discovery to drug in the clinic, and with cheaper, more effective drugs. We don't know if we'll succeed, and we don't know if we'll convince others to follow our lead, but we're optimistic, and I think at the moment that's all we can hope for.
Yeah, for sure. I think it's a good way to look at it. And I think optimism is going to get you a long way, to be fair, because what you're doing is fantastic. And thank you for going into so much detail about it. I think for me, and hopefully everyone listening as well, it'll be fascinating to sort of follow your path and see how you get on. It'll be brilliant to see what you guys can do. And hopefully you can, reach the aspirations that you've been reaching for. So yeah, thank you for coming on and chatting a little bit about what you guys do and a bit about your background. It's been absolutely fascinating.
Absolutely. Thank you so much. If I could maybe add one more thing quickly as we close out. Today seems like a fortuitous day for us to be having this conversation. I woke up this morning and looked at my phone as my tech stocks across the board were kind of dipping with the introduction of DeepSeek over the weekend. DeepSeek's kind of success is actually very illustrative of what we're trying to do at Tessel. It all comes down ultimately to using responsive feedback to drive great performance. DeepSeek was able to train with much, much less infrastructure, with much, much less data, because they leveraged that. The difference in our case is that we are actually collecting the data as we go, not just pulling from a big pool that already exists. But their success actually, I think, points the way for what we're trying to do in biology. We think that biology is poised for a DeepSeek moment.
Yeah, perfect. Well, we'll definitely keep an eye on it. I think that's fascinating. I'm going to look a little bit more into that as well after this, because I think that sounds really interesting. So, but yeah, fantastic. Thank you for that. And yeah, hopefully you've had a good time having a chat. And yeah, it'd be great to keep in touch.
Absolutely. Thank you, Emily.
Thanks a lot so much. Thanks so much, Arvind. Bye. Ooh. Thank you for tuning in to this week's episode of Vision to Venture. If you're listening, make sure to give us a 5-star review. And if you're watching, like and subscribe to listen to more inspiring stories from tech leaders.